Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 2. This is lecture number 9. The topic is Law of Segregation. This will be the second and final part of the lecture. Discussing agenda for the topic will be Mendel's model, useful genetic vocabulary, and the test cross. Now, Mendel's model. Mendel developed a model to explain the 3:1 inheritance pattern that he consistently observed among the F2 offsprings in his P experiments. So, in all the crosses, the monohybrid crosses, the F1 offsprings exhibited the phenotype of the dominant parent. When the F1 hybrids were self-fertilized with each other, the F2 progeny was produced, and in F2 progeny there were 3:1, that is 3 individuals exhibiting the phenotype of the dominant parent and 1 individual exhibiting the phenotype of the recessive parent. Now we describe 4 related concepts making up this model, the fourth of which is the law of segregation. So first, alternative versions of genes account for variation in inherited characters. So genes are the functional copies, discrete hereditary units, or these are the functional units of DNA of genetic material, and genes exist in pairs. The two members of the pair may be alike and we call them homozygous, or they may be dissimilar, then we call them heterozygous. So these two alternative versions of the gene are called alleles, and the alleles may differ from one another, each controlling the character to a different degree. In a particular individual, there can be a maximum of 2 alleles for a particular trait at a particular locus, but in a population there can be more than 2 different alleles for a particular location or locus. The gene for flower color in pea plants, for example, exists in 2 versions. One for purple flowers, which determine purple flower color, the other for white flowers, which determine white flower color. Now these alternative versions of a gene are called the alleles. Today we can relate this concept to chromosomes and DNA. Now each gene is a sequence of nucleotides at a specific place or locus along a particular chromosome. Because a haploid individual has 2n chromosome complement. 2n chromosome complement means that each type of chromosome is present in 2 copies. So a gene determining a particular character, 1 copy will be located on 1 chromosome and the other copy of the gene pair will be located on the another homologous chromosome at the same locus. Here you can see These are two different chromosomes, and on one chromosome the gene responsible for determining the flower color is white, and on the other chromosome the allele which is present is responsible for determining purple flower color. So the locus is same but on homologous chromosomes, and because these two alleles are different from one another, therefore they are controlling the trait to different degrees. And why the different alleles control the trait to different degrees? It is because of their nucleotide sequence. You can see here the allele which determines purple flower color is slightly different in nucleotide sequence to the gene which determines the white flower color. Now, through a series of steps, this particular DNA sequence result in production of an enzyme that helps synthesize purple pigment. So in this way, this allele controls the purple color. This DNA sequence results in the absence of the enzyme, so the color remains white. One purple flower allele results in sufficient pigment for purple flowers. So although this individual has one allele which determines purple flower color, another allele which determines white flower color, but the flower color is purple because this allele is dominant, and this is recessive. Remember, a dominant allele is one which expresses itself in heterozygous condition. A recessive allele is one which cannot express itself in heterozygous condition. And remember one more thing, that dominant is not always good and recessive is not always bad. The deafness in humans is because of a dominant allele. Polydactyly in humans is because of a dominant allele. So dominant allele is not always good and recessive is not always bad. The DNA at that locus, however, can vary slightly in its nucleotide sequence. So it is referring to the nucleotide sequence of the two dissimilar alleles. One is responsible for determining purple flower color, the other is responsible for determining the white flower color. And you also know that phenotype is controlled by an underlying genotype. So there are differences in the phenotype. Obviously there will be differences in their genotype, and these differences are in the nucleotide sequence of these two alleles. This variation in information content can affect the function of the encoded proteins and thus an inherited character of the organism. So nucleotide sequences are different, therefore they control the phenotype to varying degrees. The purple flower allele and the white flower allele are two DNA sequence variations possible at the flower color locus on a pea plant's chromosome. So in pea plant, the flower color can only be exhibited in two forms, that is purple and white. There is no intermediate. The purple flower allele sequence allows synthesis of purple pigment, and the white flower allele sequence does not, because it determines white flower color. Now, second, for each character, an organism inherits two copies, one from each parent. Remarkably, Mendel made this deduction without knowing about the role or even the existence of chromosomes. As mentioned in the previous video, Mendel performed his experiments in 1860s and in 1880s chromosomes were discovered and the process of meiosis and mitosis were studied. So that it is an example of the genius nature of Mendel, where he discussed entities called genes residing on the structures called chromosomes. He is discussing genes, and at that time chromosomes are not discovered. Chromosomes were discovered 20 years later. Each somatic cell in a diploid organism has two sets of chromosomes. That is, we refer to it as 2n. Thus, a genetic locus is actually represented twice in a diploid cell. So it means that in a diploid organism, chromosomes occur in pairs and genes occur in pairs. So for each gene there are two copies, and if a character is controlled by a single gene, then two alleles are responsible. These two alleles may be same, they may be different, but genes exist in pair form. The two alleles at a particular locus may be identical or they may be different from one another, as in F1 hybrids. If the two alleles at a locus differ, as in F1 hybrids, then the dominant allele will express itself and the recessive allele will be masked. The recessive allele cannot express itself in heterozygous condition. It will only express itself in F2 generation when it appears in homozygous form. The fourth and final part of Mendel's model is the law of segregation. The law of segregation states that two alleles for a heritable trait segregate or separate during gamete formation and end up in different gametes. So according to Mendel's model, we just revise these four ideas. Each trait is controlled by a pair of factors. The factors may be similar, they may be dissimilar. The factors stay together in a diploid individual, but at the time of the formation of gametes, the factors or the alleles, they separate from one another and then they are reunited at the time of fertilization. Thus, an egg or sperm gets only one of the two alleles that are present in the somatic cells of the organism, making the gamete. In terms of chromosomes, this segregation corresponds to the distribution. Now, similar behavior is also shown by chromosomes. In a diploid individual, the homologous chromosomes stay together, but during gamete formation, the two members of the homologous pair, they separate from one another, and then they are reunited at the time of fertilization. Note that if an organism has identical alleles for a particular character, then the allele is present in all gametes. So, if there are no differences in the alleles of a gene, both the alleles are homozygous, then all the gametes produced by that individual will have copy of that allele. Because it is the only allele that can be passed on to offsprings. The offsprings always look like their parents. This explains why these plants are true breeding. So true breeding plants are the ones which are homozygous for all the traits under consideration, and these true breeding plants faithfully transfer all the characters to their offsprings. Now take another case. If the alleles at a locus are different from one another, then 50% of the gametes will receive the dominant allele and other 50 will receive the recessive allele. Does Mendel's segregation model account for the 3:1 observed in the F2 generation of his numerous crosses? Now, for the flower color character, the model predicts that the two different alleles present in an F1 individual will separate into two gametes, such that half the gamete will have the purple flowers and half will have the white flowers. So in the F1 offsprings were heterozygous dominant. It means they had one allele which was determining purple flower color, they had another allele which was determining the white flower color. So during gamete formation in the F1 hybrids, some of the gametes carried the dominant allele, whereas others carried the recessive allele. During self-pollination, gametes of each class unite randomly. So there is a random combination of gametes, that is, one gamete carrying a dominant allele can either fuses with another gamete carrying a dominant allele or a recessive allele. So this is random fertilization. Since the same is true for an egg with a white flower allele, there are 4 equally likely combinations of sperm and egg. So these 4 combinations can be homozygous dominant, heterozygous dominant, and homozygous recessive. The heterozygous dominant can appear in two forms. When the dominant allele is shared by sperm, recessive is shared by egg, or when the dominant is shared by egg and recessive is shared by sperm. So in this way we have four combinations. There you can see in P1 generation we have one plant which is having purple flowers and another plant which is having white flowers. These are true breeding. This is their phenotype, physical outlook. This is their genotype, genetic makeup. Now these plants produce alleles, gametes. Remember, gamete carry only one copy, so these gametes fuse together, and these are the F1 hybrids. The F1 hybrids are purple flower carrying plants, and their genetic makeup is heterozygous, so they are heterozygous dominant, and then these plants produces gametes. Some of the gametes will be carrying the dominant allele, some of the gametes will be carrying the recessive allele, and when random mating occurs, so these four combinations are possible. And if you analyze their phenotypic ratio, then it will be 3:1:3 purple to 1 white. If you analyze the genotypic ratio, then it will be 1:2:1, because there are three different genotypes, and the number of individuals exhibiting these different genotypes will be 1:2:1. Notice that we use capital letters to symbolize dominant alleles and lowercase letters to represent recessive alleles. So P is purple flower color and small p is the white flower color, and it is often useful as well to be able to refer to the gene itself as the P/P, that is, it is in heterozygous form. One allele is dominant, the other is recessive. So in the F2 offsprings, what color will be? Will the flowers be? In the F2 offsprings, the white flower color will also appear, because the allele responsible for determining the white flower color will have the opportunity to be expressed in homozygous form only in F2 generation. One-fourth of the plants have inherited two purple flower alleles. Clearly, these plants will have purple flowers. One half of the F2 offsprings have inherited one purple flower allele and one white flower allele. So these plants will also be having purple flower color, because the allele for purple color is dominant. And finally, 1/4 of the F2 plants have inherited two white flower alleles. So they have the opportunity to come up in homozygous form, and they will be expressed. Now there are some genetic terminologies. Homozygous is one having two identical copies parental generation. The purple flowered pea plant is homozygous. Both the alleles are similar, that is capital P, while the white flower plant is also homozygous, which appears in F2 generation. So these are true breeding, where the character under consideration is in homozygous form. Now, an organism that has two different alleles for a gene are called heterozygote and is said to be heterozygous for that gene. Example is the F1 plants, which are heterozygous dominant. Now, self-pollination of the F1 hybrids thus produces both purple flowered and white flowered offsprings, because in F2 generation the recessive allele gets the opportunity to appear in homozygous form, and they also thereafter expresses their effect by determining white flower color. So phenotype is the physical outlook, it is the physical appearance, whereas genotype is the genetic makeup, and the phenotype is controlled by the underlying genotype. So here this PP and this is the genotype, genetic makeup, whereas the phenotype they determine is the physical outlook. So finally we discuss the test cross. Now what is test cross? Now we have a problem. Although the F1 hybrids are exhibiting the phenotype of the dominant parent, but we don't know anything about their genotype, whether they are homozygous dominant or they are heterozygous dominant. So how we will know the genotype of the F1 hybrids? For this purpose, we will cross the F1 hybrids with the homozygous recessive parent, and then we will analyze the outcome of this cross. When the F1 hybrids are crossed with the homozygous recessive parent, then these two results will take place. All the flowers will be purple. If all the flowers are purple, then it means the F1 hybrids are homozygous. If 50% of the flowers are purple, that is exhibiting the character of dominant parent, and 50% are exhibiting the character of recessive parent, then we will conclude that the F1 hybrids are recessive. F1 hybrids are heterozygous, as you can see here from the cross. If the cross between F1 hybrids and homozygous recessive parents results in the flowers all having purple color, then the F1 hybrids are homozygous dominant. If the cross between the F1 hybrids and homozygous recessive parent results in 50% of the plants carrying purple flowers and 50% of the plants carrying white flowers, then it means that the F1 hybrids are heterozygous. You can see the relevant text. Thank you very much. Allah Hafiz.